Thank you, Sally. And I would like to invite you first to share your research experience. And Sally, floor is yours.
Excellent. I'll just share my screen again. This one. Okay, so you can see my slides now, I'm assuming.
Perfect.
Excellent. I'll just get my notes up as well because I don't have everything in front of me. Excellent. Get rid of that. So thank you so much for the opportunity to talk today. It's currently 7:30 p.m. on a Wednesday night, so apologies if I refer to evening time frames. Thank you again. My name is Sally O'Neill. I work for Turn Ecosystem Surveillance and I'm the operations manager and I'm based at the University of Adelaide. Australia membership on Ilter is maintained by Turn for the benefit of all Australians and the Ilter network. Of the 4 thematic themes common across Ilter networks, we focus on biodiversity as well as carbon and nitrogen cycles, but not so much the eco-hydrology aspects. As the name suggests, Turn is the terrestrial ecosystem research network, and we focus on terrestrial ecosystems and don't delve into marine or freshwater systems. Right. Firstly, I would like to acknowledge the traditional owners and custodians and their profound connection to land, water and biodiversity and culture, and pay our respects to their elders past, present and emerging, not just here where I am today on Kaurna country at the University of Adelaide, but also across the landscape where Turn operate and where all Ilter networks operate. To also acknowledge our funding body, just like many of your programs, we're federally funded. Turn is funded through NCRIS, the Australian Government's National Collaborative Research Infrastructure Strategy, which is funding for research infrastructure in Australia. Just as many of our networks, which you're probably familiar with, we rely on partnerships with universities across Australia to implement our programs. So, firstly, another introduction to Turn. Turn is Australia's land observatory. We deliver ecosystem research infrastructure for the global community. Ecosystem monitoring is fundamental to our understanding of how ecosystem change is impacting our natural resources and is vital for developing evidence-based policies and management. Landscape monitoring is conducted over large areas. As you can see, Australia is quite large. We provide spatial data enabling questions relating to where and when ecosystem change is occurring to be addressed. So for take-home messages, Turn provides freely available open access ecosystem data for all. It's easily downloaded directly from our website and access is also available through our R package. Get in touch if you would like further information. We will provide the tools to support the discovery, analysis and reuse of data. We have site-based instruments and monitoring across a network of ecosystem sites and sensors. And key to my particular work is we provide method standards and protocols for field collection to facilitate research, education and adaptive management for our natural resources. And that's where I'm going to try and focus today. So the funnel diagram. So Turn operate at 3 different scales. Internally, we refer to them as platforms. I work for Turn Ecosystem Surveillance. We inform on the types of different questions that are routinely posed for ecosystem monitoring programs. So where and when change is occurring, which is typically at the continental scale, what change is occurring and what is the direction and magnitude of that change, the regional scale monitoring, and then why the change is occurring. So that's more of the local level monitoring, which for us includes our flux towers. So focusing at that regional scale monitoring is the purpose of today. So the regional scale ground-based monitoring. So it's our team going out, collecting ground data. They're taking samples, they're taking specimens, they're digging holes and bringing back the samples for everyone to use. So a summary of Turn's approach. We currently have 2 volumes of published field protocols. Our standard protocols was released in 2012 and is currently undergoing a bit of a review. Second to that, we also have a second set of protocols, which is designed specifically for tall eucalypt forests and is probably quite specially designed for Australian systems. These protocols were developed and approved by a group of scientists, land management and government staff who collaborated and worked together. It took a number of years, but we got there in the end, and we've been operating now really well for the last 10 years. We also have a suite of protocols in development that's comprising of around 24 additional modules. I'm going to get to these in a minute because that's where the exciting stuff is. Our plot-based methods have been tried and tested and widely implemented across Australia in a range of environments. I probably haven't even mentioned, but we have 835 plots across Australia. Most of them have only been sampled once, so we're going for the long-term data, aiming for 10 to 12 years of the repeat intensity. But there are 200 or so that have been done a little bit more intensively, so that's good comparable data. Our focus is on collecting quantitative vegetation and soil data rather than the qualitative observational information. Our data is supported by the collection of actual soil samples and vegetation vouchers. So it's just not data. Onto our field sample library. I hope there's some good soil scientists in here because this is just for you. Just some examples of what is available to researchers around the world. From our 1 metre deep soil pits, so there's 1 soil pit in each plot, and 9 smaller, shallower holes as well. They're much shorter. We have 25,500 gram soil samples available for analysis, as well as 2,500 or so bulk density samples. So that's really important to go with the samples, as well as over 10,000 soil samples collected from the soil surface designed for metagenomic sampling. These samples either have or are available for the analysis to provide updated digital soil information for Australia. They are improving the quality of existing soil grids and adding significant new insights to dynamic soil moisture, organic materials and nutrient data sets. Onto the vegetation information. We have a lot of samples. There's over 56,000 herbarium samples available as well. That represents over 6,000 different species. 7,000 or so of them are actually located in state herbaria or our national herbarium as well. That just means that they're readily available for researchers to use or taxonomists to get really good quick access to. But the remaining are housed at my workplace at the University of Adelaide. It's a really quick and easy process of accessing those samples. We have a good application form. It just ensures that the sample is going to be used for something that we're sort of aligned with TERN's objectives, and we just gather a little bit of information and our assessment panel confirms that yes, the sample is available for use. Go your hardest. In addition to the herbarium samples, we do have 65,000 or so leaf tissue samples ready for genetic and isotope analysis. That represents 1 sample from each herbarium sample collected. And then also we have replicates of the most dominant species that were present at the plot as well. So that can enable population studies from the genetic analysis. Just some key examples of the ecological field data and how it's being used. So I could produce you with a big long list of all the publications that have come from TERN data over the last 10 years, but some more practical examples. The Australian government has adopted our monitoring protocols to assess the rehabilitation of mine closure and mine closure planning restoration activities, something that TERN never really anticipated it would be used for. We've definitely been important in discovering new weeds in Australia. There was a very little species found in the alpine areas in Tasmania recently. Our data is also used to map Australia's biodiversity hotspots, identify their climate change sensitivities, and inform strategies for their conservation of these species. Another factor that's really important for us is training and upskilling of ecologists, of students, of researchers. We train dozens of professionals every year. We have a really good program, and it's something that's been critical for undergraduate levels, undergraduate programs. You might be aware of getting outside less often, just in the lecture theatre, or these days even just via Zoom. So us turn hosting sessions where we take undergraduates out in the field and give them a really good hands-on practical experience has been lovely. I don't have a great map of all of our plots, but 835 plots across Australia. There's some gaps. If you're not familiar with Australia, some of those gaps are sort of around the desert areas, but we are working our way around the country. We'll get to them eventually. Up until now, we've mostly focused on building our network and adding new plots, and we've just recently started to moving that focus onto revisiting the plots and gathering the more temporal data. And our exciting new collaboration with the Australian Government, Department of Agriculture, Water and Environment is to develop new protocols specifically for the Regional Land Partnerships Program. These are projects that across Australia where natural resource management practitioners are tasked with restoring ecosystems. Currently, the funding period from 2018 to 2023 has provided funding for 50 service providers across 54 land management units across Australia, expending $450 million worth of project funding to benefit our environment, our farms and our communities. These projects are currently using their own monitoring methods, either adapting their own local state jurisdiction, ecological survey methods, merging some citizen science methods, or completely coming up with their own, or quite possibly doing things a little haphazardly. With this new project that DOR are funding us for, developing or co-developing with DOR a suite of monitoring programs that will be used across the country. It's a modular set building on TERN's existing methods that we've been implementing for the last 10 years and developing some new and exciting ones as well. I get very excited. We're delving into vertebrate fauna, invertebrate fauna as well, things like camera trapping and that sort of stuff. So post 2023, you can imagine that with 50 or so NRM groups or regions having their own projects dotted across the countryside, but all following exactly the same survey monitoring, it means that we're going to have a massive data set available for our long-term research community. So just an overview of what that project is. We're aiming for consistent, comparable and scientifically robust data to make decisions about managing our natural resources, improving our ecosystems and conducting environmental impact assessments. For now, the key intended use is for the Regional Land Partnerships project, but we're also designing key components for environmental impact assessments. For the standardized monitoring, we're taking a modular approach, building on TERN's existing protocols. For now, there's 18 modules in development, and I'll skip over that bit. So the 18 modules are here. I think they're fairly self-explanatory. It's stuff about gathering the site data information, grabbing floristic samples, getting cover information, basal area recruitment, and a lot of condition variables and all that sort of stuff. Please get in contact if you want more information on those, but I'm going to skip past them. Just to summarize what a module is, it's a focused topic that typically concentrates on one field measure, one survey method. It also contains a set of instructions, guidelines. It basically means that people can't leave it open to interpretation. It's so detailed, but so concise at the same time that they can't get it wrong. Another thing is that we provide the tooling, so an app, so that there's drop-down lists that go to standard vocabularies. They're using Darwin core or more modern vocabulary, so it's all very standardized. Anyone around the world, you guys will be able to pick up that data and understand exactly what it's all about. Just some quick use cases. Consulting ecologists, for example, the apps are there. It's going to make everything from the data collection really quick and easy, tells them exactly what to do. They know their expectations, they know what equipment they're going to be using. It's all there for them. and a good peace of mind for a consulting ecologist, so someone that's been doing it for a long time. It's good to know that the data is being made useful for other projects as well. It's just not a little individual project. The data that they'll be contributing is going to the bigger, wider community as well for pooled data sets. So you guys in the international community can also access it. As a researcher, it's a benefit for the researchers. They have clear-cut understanding of how the data was collected in the first place. It wasn't open to interpretation. They know what rules can apply or not apply, what little statistics they're going to have to understand to get the data correct. For land managers, there's a whole benefit there. I'm assuming you guys aren't land managers, but I can talk about that at some other time. Just a side note on other factors that are important for TERN and how our data and samples are being used. There's also lots of learning opportunities available through TERN. We've developed a TERN data for students document and a data set to use as well. Some other examples, particularly, I guess, during COVID times, is universities from across Australia. I'll use one, for example. Monash University had 460 students accessing TERN-derived data through our TERN tooling and online systems to basically run a field lab from their own home during COVID, where they just couldn't get out in the field. So that was a really good, exciting thing. I'll just put up some quick slides on all the people that we work with closely. If you would like to get in touch, my contact details are down the bottom, and I'm more than happy to answer some questions afterwards.
Thank you, Sally, for your informative presentation. I think I got one question from Andres. How do you monitor soil temperature and moisture at a regional scale? Sally?
That's not my forte at all. I know we do it. We have sensors. There's a network of flux towers across Australia, and there's a big sensor system. It's all over the top of my head, but I'm more than happy to chat at some other time and put you in contact with the right people.
Okay. So anyone want to ask questions to Sally? Please, you can write down. Hide.
Hi everyone, I'm Hideaki Shibata from Hokkaido University, Japan, and chair of IOTR. Thank you, Sari, for your very great presentation, and I'm impressed so much about the data availability and sample archives in the different places. Especially, I am soil scientist and biochemist, so I am so impressed that the large number of soil is available for the under analysis. So my question is that soil might be collected in different places in the Australia, and do you have any repeated samples over the long time series in the same site? That'd be another good resources to understand long-term ecological and socio-ecological dynamics in that sites. If that sample is also available?
We're definitely aiming for that. TERN itself has only been collecting for 10 years. So 10 years on your terms is quite small. I understand that. It's something that we're aspiring to. There's probably some other soil archives. The CSIRO have a soil archive where that sort of information may be available, but internally it's not through TERN.
Okay, thank you. So we found many other 2 questions in the Q&A box. Amit, please. Amit, you're muting.
Yeah.
Sorry, my fault. Yeah, so Stephen said, thanks for your great talk. Regarding soil sample, are you archiving the soil sample to monitor long-term changes?
Yes, that's the intent. Yeah.
Okay, I think this is the short answer for this question. Okay, so I got another one. Given funding is always limited, how do you see the right balance between spatial coverage and being able to continue long-term at the same sites?
Yeah, it's always the question, isn't it? We receive funding from the Australian government. It's not a long-term. We don't get a 10-year budget. We get that 5 to 7 year budget. We're about to undergo another funding round. I think the Australian government through NCRIS has committed to research infrastructure. I think there's no doubt or no question about that, and they're doing a fabulous job. It's an exciting time to see what that next budget cycle will look like, but everything looks promising.
Okay. Yeah, thank you for answering. Anyone, because we have very limited time, so if you have any questions, so please.
May I one more?
Yeah,
sorry.
Please.
So the soil and plant samples, are these samples available for the international scientists like working in Japan? How can we apply to use that samples in the Australia term if I want to use?
Sure, you are more than welcome to fill out an application form. If you go to the turn.org website, there's a sample library thing, and you can follow an application form. It's quite a straightforward application form. Someone with the credentials of yourself wouldn't have any problems, and would need to look in some biosecurity if there was any treatments required to the soil before we export it and that sort of stuff, but it's all freely available. As long as you don't ask for 300 different samples and expect them tomorrow, it's all doable really easily.
Great, thank you so much. That's very valuable for the international collaborative study. Thanks.
Yeah. So Sally, do you want to add something which you want to share with us?
I'm disappointed there was no fauna or invertebrate questions, but thank you for getting on team soils. That's fine.
Okay. So I think I got one more questions from Mark. May I ask you? So how do you develop the standardized observation and protocols according to the research question to feasibility to relevance, or what was then already anyhow?
Sure. That's going to require a bit of thought. I think the key point is that we don't have the research questions. We don't set the research questions. We understand what data and samples researchers will want and need to be able to answer their own research questions, if that makes sense. So we standardize everything. We really clearly document how everything was collected and how it wasn't collected, so that there's no need for just assuming things. So yeah, it's up to the researchers to come up with the research questions.
Yeah. So okay. Like just a general questions, like Hide said, how would the opportunity for master and PhD students from outside of the Australia want to collect the data from Australian network site? Is it possible for them, or they need any process
to come to Australia, or to
to come to Australia to collect the data from the ILT like Australian network site?
Yeah. So, we're very keen to collaborate with any researcher, particularly the PhD students or postdocs. That's fine. And I can't show you anymore because I'm not screen sharing, but there's over 20 or so universities across Australia that we collaborate with. We have researchers out there. I probably didn't explain that TURN is a research infrastructure group. We don't have our own PhD students as such. We do have co-located students and that sort of stuff, but definitely get in touch if you have an idea for a project that you're interested in, or you're an existing student or researcher at an international university and go, hey, I really want to get in touch and collaborate with you. That's completely fine too. Yeah, thank you. Your information. So yeah, Sally, thank you for your sharing the presentation about term approach, how to collaborate the international, how to collaborate with other network, and also how to providing the opportunity for students. I learned from your presentation many things. So once again, thank you for your time and great informative presentation. So I think, thank you once again. I think this is the time to move on for our welcome to Nathan, our second presenter from Israel. Nathan, if you don't mind, could you please share your presentation and let's get started.
Of course, I will share my. Okay, everybody can see it?
Perfect.
Okay, so as I was saying, my name is Nathan Levy, and I'm a PhD student at Ben Gurion University here in Israel, but also in collaboration with the Agricultural Research Organization and the Volcanic Center of the Institute of the Ministry of Agriculture in Israel, under the supervision of Professor Arnon Carnelli and Dr. Saril Paz-Kagan. And today I will talk more of a study case, unlike what Sally presented, more of our study case of using airborne imagery spectroscopy to assess the land use effect of soil quality on soil quality in dryland regions. So, as mentioned, Israel it has very different types of climate types, but desert occupies about 60% of it, and I will show the different mechanisms of land use effect on the soil quality using airborne imagery. So it's not a big shock that the global population is growing rapidly, and this requires a lot of resources and systems, like ecosystem services, to sustain this growth. And for that, we need to produce more food and more of these services. And this requires a lot of transformation of natural ecosystem and soils into human-dominated ones. And this can, when it's not done properly, this can turn to be a deteriorated soil quality. And for that, we need to develop and to apply different techniques to monitor this soil quality for a sustainable growth. There are many different types of hazards that blur the quality of the soil, especially in desert area, because there's such a big lack of water and organic resources availability to the soil. It could be from hard pressure of human activity like agriculture and grazing, but also the imbalance of water and organic resources availability that can deteriorate the soil quality by sedimentation or for growth of desert vegetation. And this needs to be monitored very strictly. So this whole approach is under one roof of soil quality assessment. The soil quality, as mentioned, is very important because it supports not only human life, but also the entire ecosystem that is under this area, and the soil sustains them all. It could be starting from animals to cropping to vegetation and other microorganisms that reside in the soil. And therefore, we want to preserve their quality and their carrying capacity, not only for us, but for the entire components of the ecosystem. There are many different types of soil quality assessments, and some of them are more focused for agricultural purposes by the Cornell Comprehensive Assessment of Soil Health. But in our case study, we use the Soil Management Assessment Framework, the SMAF, which is more flexible in terms of selecting more appropriate soil indicators that best depict the soil condition of the specific study area that we study. So there are many different types of soil. What is even soil quality? It's kind of an abstract term. So in order to measure and to quantify the soil quality, we need to use quantifiable, measurable soil indicators that are usually divided into three general groups of physical, biological, and chemical factors. Each correspond to a specific soil mechanism and soil characteristics. There are different types of them, dozens of them, and you can't really measure them all because it will create a very complex assessment model. So if we want to be more precise and more specific about the study area, the soil in the study area that you want to study, you need to select the very exact and very considerate soil indicators that you want to incorporate in your research, and this is exactly what the SMAF is doing. So soil is a very complex system, and there are many different types of mechanisms that affect its quality and health and performance, and many of them are also affected by the soil texture. Soil texture is a fundamental property of the soil, and compact soil will retain more water and will allow less percolation and infiltration of the water, and the solutes and the different materials in the water solution to go through the soil column, whereas an aerated and very coarse soil texture will allow more infiltration. And this is very important for the soil because if there is more water and more organic resources available in the water, many of them will eventually retain to the fine particles of the soil. The clay is negatively charged and it attracts and absorbs a lot of materials that are in the water, like different types of elements, organic matter, also heavy metals and other materials that are in the soil. And this heavily affects the quality of the soil. If the soil has a large pore area, it can absorb a lot of these materials and improve its quality. And if the soil is very coarse, the soil grains are very coarse, it will not retain many of them, and this really deteriorates the quality of the soil. In this desert area, in our study area, it's mostly desert. It's very rich in sand. It's mostly located under the sandy loam textures. So there's a relatively small fraction of clay minerals and clay texture size, and therefore the soil is not rich and not available for a lot of these materials. Therefore, the soil is not very fertile, but still we can use them to measure and to see the different concentrations of the different materials in the soil. So not only by the measurements themselves, this is where spectroscopy is coming in, and this is what we are doing, introducing remote sensing into this approach. And when looking on soil spectroscopy, we can detect the different concentrations and different different materials in the soil based on spectral signatures that we collect both in the laboratory and in the field. This is a typical shape of soil spectral signature of a soil, and you can see that in different spectral regions. In this case, we focus on the visible near infrared and shortwave infrared spectral regions between 400 and 2500 nanometers. We can see that different spectral features. You see those little valleys or specific reflectance, higher reflectance that corresponds to the presence of different soil materials like organic matter and clay minerals and water in the soil. We can see the difference. So each of the soils have a relatively similar shape of the spectral signatures, but based on the different concentrations and conditions of the soil, there will be shown differences in intensity or in specific spectral features. And based on this concept of the difference, we can differentiate between soils and the materials in the soil, and we can look at it not only on the point scale but also on larger extent. So remote sensing, when looking, the concept of remote sensing is to acknowledge not only at the point scale but looking at a much larger extent, and we can use that in different platforms. It can be from drone imagery, airborne imagery, also satellite imagery, but we usually split these two into. We want to acquire as much spectral information as possible. And in this group, there are two major groups of sensors. We have multispectral images, sensors, and we have hyperspectral images. The multispectral is very robust, it's very confident, and it's quite, it's very well known in the science of remote sensing, but we only acquire very few spectral bands. We can get an information that is not given, it's not deep enough to get all the spectral features that we can collect that we have seen in this type of signature. So therefore, if you want to acknowledge on a larger scale the very subtle spectral features, we would use imaging spectroscopy that is based on hyperspectral imaging that contains hundreds of spectral bands, and each of them can give us a very specific absorption features that we can then infer on the presence of the different materials in the soil and create this large extent pixel-based imaging of the spatial distribution of the different phenomena that we want to map. So as mentioned, the main objective was to study the effect of the anthropogenic activity on soil properties and soil quality in the ecosystem, and more specifically to see how imaging spectroscopy can adhere to this goal by incorporating both the laboratory survey analysis and the spectral dimension. and also to see if we can use this approach in arid conditions that are very poor in the availability for the different limiting factors like organic matter and water availability. This is a general flow chart of the study. It's divided into 3 major steps. The first one is the soil collection and analysis, and eventually also the calculation of the soil quality index, which integrates all the individual soil indicators into one comprehensive index that gives you a score between 0 to 1 to show the performance of the soil. The second step is mostly referred to the imaging itself. So we have a flight campaign that took 3 strips of images with a scanner mounted on a light aircraft. We'll get to that. The whole correction and pre-processing process of the image itself, and then we use the spectral information from the image to construct a regression, and the imaging, basically the imaging spectroscopy analysis itself, combining those 2 data sets to eventually map on a larger scale, on a regional extent, the different soil properties and the SQI itself. So a bit about the station itself. This project began as part of the EU, European Union 2020 Horizon eco-potential project that monitors different types, a large variety of types of climate types, including Har HaNegev here in Israel, and of that region, which is one of these stations in Har HaNegev. It was established in 1958, and it corresponds a lot of the human influence on the ecosystem viability, because this, and also on the effect of runoff events and runoff harvesting for agricultural purposes, as this is an arid region. It is highly affected by seasonal raining, especially in winter time, that causes different runoff events. So this is our study area of that region. You see here, it's located in southern Israel, where deep in the desert area of the Negev, and this is a sparsely populated region, but it still contains different types of human activities. We have the natural ones, but we also have the agricultural, mostly of single-family farms for vines, for winery, and for olive cropping. We have the nomadic Bedouin villages that mostly adhere for grazing purposes of livestock and sheep. And we have the RHS, which is the runoff harvesting systems. We can see the different types of land uses. So as mentioned, we have the natural land, we have the local farms, agro-pastoral grazing, and we have the runoff harvesting system that is comprised of 2 different types of water catchment systems. The first one is called demand, which is a hedged catchment water system that is used for small-scale forestation and agriculture. And the other one is the stone wall terraces that usually is used to reduce the energy of the water flow and also retain a lot of the essential organic resources that are transported with the water and washed up from the entire region into the stream system. This is a general overview of how to construct a soil quality index approach, soil quality assessment. So as mentioned, we collect the biological, physical, and chemical soil indicators, constructing the minimum data set. Then we go through scorings and transformations, so they can be compared between the different types in a unified scale. And eventually they are integrated into the soil index. It could be soil health index, soil quality index. The concepts are quite similar. There are many different types of approaches of how to do this. So I will show you exactly what we did in our case. So our properties, we selected 12 of them, physical, biological, and chemical ones. So we focused on the most limiting factors of our study area. We use, of course, the water availability of the soil and the soil organic matter, including, of course, the availability for soil nutrients like nitrate, phosphorus, and potassium. But as this soil is naturally more saline than others, and the lack of water availability can really create salinization processes in the soil. We also monitor different types of saline elements and electrical conductivity, which are essential to monitor the soil quality. The next step was, of course, the laboratory analysis and to acquire all the measurements of the soil. And then the development of the soil quality index, as mentioned, was first started by I'm sorry, to transform the original values into a comparable scale between scoring between 0 and 1. And we use 3 different types of transformation. We have the more is better, less is better, and optimum. The more is better grants higher scores to soil indicators that generate and improve the soil quality, like water availability and organic matter. On the other hand, we granted lower scores for higher concentrations of soil degrading materials like salinity, like saline elements, electrical conductivity, and some others, and the rest were generated in optimum scores for properties that we wanted to keep a more balanced mechanism. And the next step was to rule out intercorrelations between soil indicators to prevent redundancy in the model. We want to be able to see what is the contribution of each of the soil indicators, and we saw that the salinity indicators were highly intercorrelated. So therefore, we ruled these 3 out, and we left only the electrical conductivity in the soil quality index calculation. Then this was followed by performing a principal components analysis to generate the weights, the weighting factors for each of the soil indicators, were eventually were multiplied by the transformed scorings of the soil indicators to generate the soil quality index value for each of the collected soil samples in the field. And this is giving us the ground truth, the reference, so that we will use later on for the large scale prediction. So this is very accurate for the point scale, but we want to move to a higher extent. So we got to the spectral dimension. So as mentioned before, we use a hyperspectral sensor for the imaging spectroscopy process. In this case, we use the isotonic sensor. It contains 420 bands between 380 to 2500 nanometers, with a spectral resolution of about between 3 nanometers to 12 nanometers for each of the wavebands. This means this is a very accurate, and we can see the very specific changes of the spectral features in the soil samples. The sensor was carried on a light aircraft and took 3 stripes above the study area that was then had to go through a very long process of pre-processing, different corrections for atmospheric scattering, for radiometric correction, and also to be geo-rectified one next to the other. And then at the end, they were unified into one image that could be used for further processing. And the next step was to clean out, to extract to remove all the pixels that are not relevant for this research, meaning all the non-soil pixels. So we calculated the normalized difference vegetation index to exclude all the vegetation pixels. This was followed by a land cover classification process, also to find out other road and built-up areas of soils that were not relevant, and also for very bright soil because they refer to chalk, very unconsolidated and degraded soil that did not represent healthy soil. And also, unfortunately, in this case, we had to, due to bad interpretation, bad classification on the edges and the fringes of the classification, we had to clip out and remove the very extreme fringes. So this is the clean hyperspectral image that we used in the processing. We removed some of the noisy bands as well due to water absorption regions in the spectrum. So we remained with 336 spectral bands. that are relevant for the rest of the spectral analysis. And here what we did, unlike other studies that usually spectrally measures the soil samples in the lab and use these measurements for high scale prediction, we use the spectral information from the image itself. These locations are the original locations of the soil sampling that we took in the field survey, and we took the 9 pixels surrounding these pixels and enriched the spectral information we collected, and this way we can address directly to the image itself and not making only this transformation from laboratory measurements to the image, but from we collected the spectral information, we extracted it from the image itself. And eventually, to perform this regression of the 2 different data sets, we used a support vector machine regression approach. Of course, there are many different types of regression methods that could be used. Some of them are more advanced than others, but support SVM is a very reliable and well-known approach in remote sensing. So we used this one in our study, and we found good prediction. But before we ran the regression itself between the 2 data sets, we wanted to see the spectral differentiation, the ability to differentiate between the different land uses that we studied in this study area. So these are the mean raw spectral signatures we extracted from the image itself, and we see only by looking at it, we see significant differences between the different land uses. The spectral shave of the natural area is much higher because there is less organic matter and less chemical components in the soil that absorbs it, that more absorbs the spectral signature. As I said, it's a sandy loam, so it's very rich in quartz. The spectral reflectance is much higher than others, where in agriculture, usually the soil is mostly irrigated and is being fertilized and composted with organic matter. So this naturally makes the soil darker, and we see also very significant spectral features of the soil. And we also ran the principal component, the partial least square discriminant analysis to create a classification between the land uses. And we saw a very good differentiation between the classes, which is a very good indicator for further regression process. Also, what the partial least square discriminant analysis generates is the VIP, the variable importance in the projection, to see which of the wavelengths, which of the spectral feature is more prominent for each of the land uses. So we see that the agricultural land uses is very strong in visible region of the spectrum, which corresponds to organic matter usually, and we can see it also for the grazing because it has a very intense herding and livestock activity. Whereas for others, we saw a lot of spectral features in the shortwave infrared region that is mostly correlated to calcite and to clay minerals present, and we will see that also later. These are the results of the soil quality index. We see the different land uses. We see that the agriculture land use has the highest because it's well maintained compared to the others. It's well irrigated and then fertilized. Also, the grazing had a lower one because it's less regulated as the agriculture. But we saw that the biological components of both agriculture and grazing are significantly higher than the other more natural land uses, like the natural and the runoff harvesting systems, because they are reliant on the accumulation of naturally occurring organic resources, whereas these two are artificially increased by human activity. We saw also significant differences for the chemical components, for the other soil nutrients and salinity indicators as well. So this really shows us the difference between the mechanisms affecting the soil. And eventually, we used this to measure the ground truth measure soil quality index, and we performed a regression using the spectral information we extracted from the image, and we saw a very good correlation between the two data sets of the measures, and it's predicted based on the spectral information. We reached a validation data set of 86%, which is very good, of course. And we see a good correlation for all the range of the SQI scores. And we use this model to eventually predict it on the large scale on the image itself. And this is the eventual soil quality index map that contiguously map the entire study area of the image. And we can see some very clear trends in this soil quality index map. We see that, for example, the stream channel, the scene stream that runs through the study area usually presents higher soil quality index values because it acts as a sink of the different soil materials, so organic materials and water and other nutrients as a sink, and it eventually runs through this stream path and improves its quality. We can see that you see it as well here in the Bedouin villages of a grazing site. We see these little hotspots of degraded soil quality because, as I said, there's a very exceeded concentration of organic material and defecation and livestock activity in an unregulated way. So this really harms the soil quality, but the in-between areas that are less affected, we see this gradient of soil quality that improves as you go far from these livestock pans and concentrations. Here in the agricultural fields, you can see the different, you can see a changing trends of some of the fields that were looking presented better SQI scores, where the others, where they are less well maintained, shows lesser effect. And this is one of the runoff harvesting systems that we also observed, and we saw that in general it presented a pretty good SQI scores. So this really, you can really see the different mechanisms that affect the soil quality, local topography as well. You see here, this is in here you can see that the soil column is much less developed because it's on a hill slope, and we see as well right here that the SQI presented is much lower. So you can clearly see the different soil degradation processes and the soil formation on the other hand. And this was also evident for the rest of the soil quality of the soil indicators. Not all of them were successful. We managed to predict, on top of the SQI, we managed to predict seven other individual soil indicators, notably the fractional sand and clay, the soil organic matter, the electrical conductivity, and the phosphorus and soil absorption ratio. And we clearly see the effect that the human activity has on these soil indicators. We see for the organic matter material, for example, that in the agricultural field, it's hard to see in here, but when you get a closer look at it, you can see that the stream bed itself collects a lot of this organic matter and improves the soil quality of the soil, but also in the agriculture and other types of land uses, we see changing patterns of the organic matter. The same way for soil nutrients, we saw that where there's intensive cropping or intensive grazing, we saw much higher concentrations of the different salinity and nutrient indicators, the concentrations in the soil, referred to the natural land, which is much poorer in those elements. But all in all, we got to see a very clear and very good interpretation of the different distribution of these materials spatially.
Nothing. Sorry to interrupt you. Could you please conclude, because we have only 4 minutes?
Yeah. This is really two less slides.
Thank you.
Really at the end.
Thank you.
And eventually, this is the spectral inference that we saw earlier. So this was done more on soil property basis. We saw we see how each one of them corresponds with the spectral signature, and eventually we saw they also coincide well with the soil quality index that integrates most of them. This is just a summary table for all the numbers. We saw we can see here the most notable spectral features, and also on how they behave for the prediction. In conclusion, we managed to show that the imaging spectroscopy was a highly viable approach to make this soil quality mapping, soil quality assessment mapping on the larger scale. Hyperspectral imaging was highly sufficient for this task. We saw the different mechanism of soil formation and soil degradation processes, and how the human activity and land use has affected these mechanisms. And this tool was found to be highly accurate, not only for the larger scale, but also in the near future. We believe that it will be more applied in different study areas and sceneries, as a new generation of hyperspectral satellites are starting their first steps in orbit, and will be more available for future studies. And as I mentioned, we presented it as a paper, and it was just accepted. It was accepted for publication, and it should be published any day now in the ICRS Journal of Photogrammetry and Remote Sensing. And hopefully you have questions, and thank you very much for listening.
Thank you, Nathan, for your informative and great presentation. Actually, we have I think,
very little
2
time.
minutes. So I think I cannot take any questions, but thank you once again for your informative and great presentation, and congratulations for accepting your paper in the Remote Sensing Journal. That is really
thank you very much for hosting me. It's been a pleasure.
Yeah, it is really great. And I think this is the time to say thank you to both of you and our participant who joined our today's webinar during this challenging time. So thank you once again, both of you and our participant. And I think soon we will upload this presentation in our ILTR YouTube channel. So you can watch again and again. So thank you once again. And I think this is a time to say thank you to everyone.